Good morning. This is Miss Degenford, and we're going to be examining our cells. We're going to be starting our cell unit, and we're going to start out by looking at the difference between the different types of cells. And as we do that, we need to do a quick review of the measurements, the metric system. And some of these units should be familiar to you, so let me scroll down here a little bit. And you'll notice we're starting in meters, and of course we have organisms, the human, and some cells, like the nerve and muscle cells, are extremely long, and so they can approach a meter. Those are going to be the ones that are found in large muscles or nerves along a limb. And then you get down to the chicken egg, and we're starting to get into the centimeters and 1 millimeter. So those we can see without the microscopes. Then we have to get into light microscopy, where we can start to see the human egg, the plants and animal cells, and then down into the micrometers, nucleus, most bacteria, mitochondria. And before we move on, let's just quickly review. Um, we might remember from previous classes 100 centimeters, or just to put it in exponential form, 10 squared centimeter, that's going to equal 1 meter. 1,000 millimeters is going to equal 10 to the third millimeters. And then that equals 1 meter. And then the next level down, which you have heard before, but probably needs reviewing, 1 million, and then we get to a unit called micrometers, and we use this little Greek letter mu to represent the M in micrometer, because we can't have another M. So 1 million micrometers, so that would be 10 to the 6 micrometers, is going to equal 1 meter. And then if we keep going, 1 billion nanometers is going to be equal, and of course that's 10 to the ninth, nm, that's 1 meter. And you can even go down, there's another one, 10 billion angstroms. But these are going to be our most useful measurements for our measurements in length. Now, all of these equal a meter, so then you can do some useful conversions. For instance, you can say 1,000 millimeters is equal to 1 million micrometers, and of course that's going to equal 1 because they are equal to each other. So then you can reduce that down. 1 millimeter equals 10 to the third micrometer. So it's going to be useful when we start estimating the distance of cells in the microscope, understanding just the comparisons. It's good to start having a sense of it. You're not going to need to memorize these, but the more familiar we are with the sizes of things, the more ease we have of understanding how they work. So, I'm not going to test you, and you're not going to be tested on a question of, well, an egg is so many micrometers, but you need to have a sense of, does this make sense? And bacteria are going to be smaller and the eukaryotic cells are going to be bigger. Once we get past the 1 micrometer, it's going to be harder and harder to see things well with a light microscope. You can see bacteria, but not really that well. Stronger light microscopes are going to see better. So you can see we might be able to get into the smallest bacteria and viruses. But at that point, we really are going to be looking at microscope pictures from electron microscopes. And you have the scanning electron microscopes, which can see the surface of images, and then the transmission electron microscopes, which, similar to light microscopes, you kind of see a two-dimensional view of the internal structures. All right, so let's start looking at the different types of cells. Now, here we're looking at a prokaryote. And over here, we'll put some comparisons I can't write today, for a eukaryotic cell. And the carry part, the eukaryote, that's the nucleus or the central part. And the pro and prokaryote, that's kind of referring to before. So you might recall from previous classes that prokaryotes have no nucleus. Eukaryotes do have a nucleus. And then we start getting into other differences. Well, you might recall, think through in previous classes that prokaryotes in the cytoplasm, they have no membrane-bound organelles. So no nucleus, no rough ER, no Golgi apparatus, no lysosomes, no organelles have a membrane surrounding them. This also is related then to its size. So this is much smaller. The very, very smallest bacteria range from zero point 0.1 to 1 micrometer, but more typical, more average sizes are 1 to 5 micrometers. Again, you're not going to need to memorize these, but it's good to have a sense of comparison. So it helps you put things in perspective. You're not surprised by things. Bacteria are going to be small. And then if we keep going here, eukaryotes, well, they do have membrane-bound organelles. They have quite a lot. Now, what you should be thinking of is why do we see these? Or the other related question is, how is this going to help with the eukaryotic systems? Why do they need it? And it is going to, again, be related to the size. It's going to be much larger than the prokaryotic cells. So these are going to range 10 to 100 micrometers, and you're going to be doing a microscope lab that is going to ask you to estimate these cells. And so these numbers should come, you should be able to figure them out, and you'll be able to verify, yes, they are going to be around that size. And then let's see, what are some other differences? Well, because they don't have any membrane-bound organelles, that changes how they do some metabolic activities like transcription, translation, how do they make proteins. Now, are there some things that eukaryotes and prokaryotes have in common? Well, imagine this is a Venn diagram, and so if I were to overlap these, well, they both have DNA, they both have a chromosome. Now the prokaryote, the chromosome is a circular chromosome, whereas in the eukaryotes they have linear. They are just one long strip of DNA. And we'll get in more detail later on with that. Well, what else do they have in common? Well, prokaryotes and eukaryotes both have ribosomes, which are going to be the structures that make proteins. Now, there are going to be some differences in their molecular structure, but they both have them, and they both make proteins for it. And they, of course, both have a cytoplasm that contains enzymes that are constantly undergoing chemical reactions. So there are some similarities. They are bounded by a membrane, both of them, and that is going to allow things through the cells, so and out of the cells. So that's what we want to kind of examine. Now, if we keep going, that gets us to the question of size. Now, just to give you a nice understanding, this picture right here, that's from a transmission electron microscope. And what you are actually looking at here, these are the two layers of the membrane, the cell membrane. So that'll give you an amazing sense of just how well those electron microscopes can work. That you're actually seeing the two layers. And this membrane is going to serve a very important function. It's going to allow substances in and substances out. Now, that's really important. I want you to imagine a stadium, a football stadium, or soccer, whatever you want to think. And so we've got this stadium, and here's where the people are going in and out. And so let's just start with the simplest. We've got a really small stadium, and the people come in and they sit down, and only one entry and exit is sufficient. They can enter the stadium and exit the stadium. But imagine if we make this a lot bigger, the stadium. And here's the field. Yay, they're playing the game, and you get all the people inside. And hopefully you can imagine that the bigger the stadium, the more people that we're going to be able to hold within that stadium, and the more trouble that they're going to have going in and out. And that's going to affect the travel. Now, we can talk about stadiums all we want, but for the cell, this means important things like oxygen, carbon dioxide, glucose. So the surface area is extremely important to the cell. So if you take a look at these numbers down here and you measure the amount of surface area, the smaller the cell, the greater the surface area to volume ratio. So for instance, if we compare a cell that only has a unit of 1, now they didn't give a like a measurement here, like 1 centimeters, but you can fill it if you want, but it doesn't really matter. Notice the surface area is 6, the volume is 1, and then the surface area to volume ratio is 6. Well, okay, so far so good. But notice what happens when you increase the size of the cell. The surface area is only a measurement that is squared, right? You have the sum of the surface areas, height times width. So that's 150. But here's something interesting. The volume is a cube. It's a cube relationship. So this increases very fast. So as this increases, notice the ratio goes down. So the larger the cell, the harder it is to get things in and out. Just like you can imagine, if you have this large stadium and only one way for people to get in and out, then that presents a problem for that stadium. They're going to have these huge jams of people getting in and out. Now, what we see with animals then, they have this interesting relationship. The smaller the cell, doesn't matter how many you have. What we can do is we can, instead of having one cell, we have many cells. And then that's going to increase the surface area and provide the needed oxygen or carbon dioxide, the molecules that they need. So the surface area to volume is extremely important, and we'll be looking at that in a lab later on. Well, I hope that help start the conversation, and we'll see you in class.